Welcome to part 3 of the biology paper 1 revision videos. In this one we're going to look at unit 3, which is infection and response. This is kind of split into two halves really, this unit. Part of it's looking at the different types of pathogen and different diseases that they can cause. And then the other half is kind of looking at the different ways in which the immune system can defend itself from pathogens. So if we just start first of all with a bit of an overview on kind of pathogens and different diseases, ties in slightly to Unit 2, but just make it really clear before we carry on the difference between non-communicable and communicable diseases. So non-communicable ones, as we looked at in part of Unit 2, are ones that cannot be spread from person to person. So good examples of these are cancers and CHD, so coronary heart disease. Communicable ones, which is what we're going to focus on in the remainder of this section, are diseases that can be spread from person to person as they're caused by pathogens. So typical examples, things like measles and salmonella. We're going to look at some more specific ones for each of the different pathogens in a minute. So the main ways in which communicable diseases can be spread are by direct contact, through water, through air, so in kind of like droplets in the air like sneezes and coughs, through vectors such as your mosquitoes, which are transmitting malaria. So that's basically those, just a couple of ways they can be spread. The list is kind of longer than that. Pathogens are disease-causing microorganisms, and there's 4 groups that we need to be aware of. So viruses, bacteria, protists, and fungi. Okay, so we're now going to look at each of the different types of pathogen and a few different examples of diseases that they cause and kind of symptoms and things like that. So, starting with viruses then, let's just move down to here. Viruses in general reproduce rapidly inside body cells, which obviously causes damage to the cell. As the viruses that have been produced kind of burst out through the cell, they usually destroy the cell in the process. Some examples of diseases caused by viruses are things like the following. Measles is a virus that can affect humans. Typical symptoms of measles are fever and skin rash. It can be spread via droplets in the air, so through coughs and sneezes. And the main way which we've kind of reduced the occurrence of measles in our country particularly is through vaccination, so vaccinating infants against measles. HIV, another virus that can affect humans. This one's spread by sexual contact and exchange of body fluids. Initially, when people get HIV, they will have flu-like symptoms, and the virus after that kind of slowly destroys the immune cells, particularly a special type of white blood cell. And eventually what will happen is without treatment, this can then progress into AIDS. And typically when somebody has AIDS, it's not actually the HIV itself that will actually cause the person to die, but it will be something else that they will catch as a consequence of having a weakened immune system due to the fact that the virus has actually destroyed their immune system cells. So quite often it can be a pneumonia or something else that people with AIDS actually die of eventually. The main treatment for HIV, because currently it's not actually curable, is antiviral drugs, and what these will do is just stop the virus replicating and kind of limit the damage that it's able to do to the immune cells. So that's why nowadays people can live with HIV for years and actually not progress into AIDS, because those drugs will kind of stop the virus replication and the destruction of those white blood cells. Our third example of a virus then is TMV, so tobacco mosaic virus, which affects plants only. So this isn't a human one, it's a plants one. The main kind of signs of tobacco mosaic virus are mosaic patterned discoloration on the leaves, and what this actually does is reduce the chlorophyll in the leaves. Therefore, the plant can't photosynthesize effectively, and it basically ends up usually with kind of a stunted growth as a result of the lack of photosynthesis, lack of glucose production, lack of energy being able to be released during respiration. So it's kind of a knock-on effect with that one. Moving on then to bacteria. So a couple of different diseases for this one. So in general, bacteria can either damage cells directly or they produce toxins which are like poisons that damage cells or tissues. Salmonella is a typical example of a bacterial infection, and this one is the one that's kind of a typical food poisoning infection. It's usually spread through the ingestion of poorly cooked food or just a general lack of hygiene. Typical symptoms: fever, cramps, abdominal pain and diarrhea. With most people it will tend to sort of go away on its own after a while. If not, antibiotics usually can be used to get rid of the salmonella bacteria. Second human example for a bacterial infection is gonorrhea. So this is an STD, so it's spread by sexual contact. Symptoms are usually a thick yellow or green discharge and pain when urinating. Typical treatments for this are antibiotics, because obviously it's a bacterial infection. Condoms can also kind of prevent the spread of it as well. So those are kind of the two methods of prevention, and then obviously treatment with the antibiotics. However, there is actually an ongoing problem with gonorrhea because various resistant strains are starting to emerge, so it's becoming harder to get rid of with antibiotics. Moving on then to group number 3, the protists. These are single-celled eukaryotes, so they have nucleuses, they're eukaryotes. The main example of protists that we need to be aware of is malaria. This one is spread by mosquitoes, which act as the vector because they transmit the malaria protist. Typical symptoms are fevers, and they actually can be fatal, so it's like a really severe fever. And the main kind of preventative measures are mosquito nets to actually stop or reduce the chances of being bitten by them, particularly at night when sleeping, or killing the mosquitoes. So obviously if you kill or reduce the number of mosquitoes, kind of using insecticide and things like that, it actually then will reduce the likelihood of malaria being spread around. Group number 4 then, the fungi. So the main example for this one is just a plants example. So we've got our rose black spot, which is in plants only. It's spread by spores carried by wind or water. Typical appearance of it is purple or black spots on the leaves, which eventually usually turn yellow and drop off early, and this stunts the growth. Fungicides are the typical treatment for this, or removing and destroying the affected leaves, so that can kind of prevent it spreading between plants and things like that. Moving on then to the ways in which our immune system can respond to any of those types of infections, or any others for that matter. We initially have a group of defences called the non-specific ones. So these are kind of generalised for any type of pathogen that might be trying to invade our bodies. So the skin is one of our best defences really for any outside pathogens, in that it acts as a physical barrier to stop them entering our bodies. Hence why if you're cut, that kind of breaks that barrier and puts you at risk of getting an infection or something like that. So skin, physical barrier. In the nose, hairs trap any kind of bacteria and pathogens that are trying to come in potentially, or be inhaled. The trachea and the bronchi have mucus secretions which then trap, and then also the ciliated cells will actually beat to kind of shift that mucus up into the back of the throat, where it can either be swallowed and digested by the stomach acid or dissolved by the stomach acid, or it can be spat out. So that's another kind of way of stopping pathogens entering the respiratory system. And then the stomach contains hydrochloric acid, which, like I just said, if bacteria make it that far, or any pathogen really makes it that far, it potentially will be destroyed by that acid. So, looking at the more specific defences then of the immune system, the main cell involved in our immune system defence is the white blood cell. 3 key ways in which they help defend our bodies. So phagocytosis is the first one, by which they surround, engulf and digest the pathogen. Number 2, the production of specific antibodies which bind to antigens on the pathogen, causing the pathogens to clump together, and then typically after that stage, phagocytes will come in, surround, engulf and digest. Number 3, production of antitoxins, which are obviously designed to neutralise the toxins that particularly bacteria are able to produce. So those are your 3 main mechanisms of the white blood cells in the specific immune response. Ways of boosting immunity then, so this is thinking about vaccines typically. If the same pathogen re-enters the body, the white blood cells will respond more quickly to produce the correct antibodies, and this is what we're thinking about in terms of people developing immunity. So this happens even without vaccines. So say you just happen to get the same infection twice, the second time it shouldn't in theory be as bad because you already have the ability to produce the correct antibodies. Vaccines kind of speed up this process in that a vaccine usually contains a small quantity of a dead or inactive form of the pathogen, and usually that will contain some of its antigens. That will stimulate the white blood cells to produce a specific antibody and therefore sort of develop immunity and memory cells for that type of pathogen. When the person is then subsequently actually exposed to the pathogen for real, the response will be much more rapid and they'll produce a much higher concentration of antibodies so that that pathogen can really quickly be immobilized and destroyed. With vaccines as well, there's kind of this additional benefit of something called herd immunity, and what this is kind of thinking about is the fact that if a large proportion of the population can be made immune to a pathogen, so loads of people in the population are vaccinated, then the pathogen cannot spread easily, because obviously if lots of people are vaccinated and can therefore quite easily destroy the pathogen, it's going to reduce the likelihood of it actually being able to successfully spread between people and infect multiple people. Final section of Unit 3 then is looking at other types of kind of drugs and treatments in general. So when we're thinking about drugs, there's kind of two main groups we need to think about. Painkillers are those that are just used to treat the symptoms. They don't actually destroy the pathogen or anything like that. It's just a case of reducing the symptoms. Antibiotics actually kill bacterial pathogens, but the key thing with those is it's only bacteria that they will work against. With antibiotics, we've obviously got this concern of the fact that bacteria are becoming resistant to them. So there's a few rules for using them, in that you should only use them if they're really needed, so it's an actual serious infection that warrants antibiotics. Don't use them against viral infections because they just won't work, and you must complete the full course. Traditionally, drugs were extracted from plants and microorganisms. So, for example, digitalis is a heart drug that came from foxgloves, the plant. Aspirin used to come from willow trees. Penicillin from mold. So that's how we first started to kind of creating drugs that we'd use to treat things. Nowadays, they're synthesized by chemists in pharmaceutical companies. So it's kind of using the principles we've identified with the traditional methods, but now we can kind of make them much more quickly en masse from the original atoms that actually make up the molecules that form the drug structure. Final bit then, drug development. A couple of different stages just outlined here. So the first stage typically in developing drugs, or once they have been developed and testing them, is the preclinical phase. So this takes place in a lab where cells, tissues and live animals are tested on to see the safety and the kind of toxicity of that drug. If that's okay, then they'll move on to phase 2 where they use healthy volunteers. It's typically a much lower dose, and at this point they're just looking to see whether on humans the drug has the potential to be safe. If that's successful, move on to phase 2 and 3. In phases 2 and 3 it does vary slightly, but in general it's patients and healthy volunteers. It's a large scale trial in that thousands of people will form part of that phase. They're looking at safety, toxicity, trying to work out the optimum dose and kind of successfully identify any possible side effects and their likelihood. And also at this point it's where it's usually done double blind, so the doctors and the patients don't know who has actually been given the drug and who has been given a placebo. If that's successful, they'll then move into publishing and peer review, where kind of fellow scientists and doctors all over the world will actually look at the results and the findings and decide if they think they're viable. And then if that's successful, the drug potentially will be licensed for kind of use by the general public or as prescribed by a doctor. So that's pretty much an overview of most of Unit 3 there for infection and response.